Hello, my name is Joshua Arnold. I am a current graduate student at Grand Valley State University, and today I'll be talking to you about my thesis project, which is assessing the short-term effects of translocation on freshwater mussels and determining if habitat or water quality is more important. For a little background information, freshwater mussels are widely considered to be the most imperiled group of animals here in North America. Greater than 70% of species are currently listed at either the state or federal level and are currently facing extinction. In addition, at least 10% of species have gone extinct in the past 150 years alone. This dramatic decline is often contributed to historical exploitation for freshwater pearls and buttons, wide-scale habitat alteration in the forms of dams and channelization, and the introduction of invasive species such as zebra mussel and round goby. A common practice to help save imperiled populations of mussels is translocation. Translocation is the act of moving a population to another place in order to spare it from being extirpated. Translocation has widely been used since the early 1900s but has varying levels of success, where some instances greater than 90% of the mussels moved during the process will survive for 5 years post-movement. In other cases, 100% of mussels will die shortly after the project. So far in this project, I have selected 4 sites, 1 control and 3 experimental sites by looking at differences in habitat using parameters such as substrate composition, water velocity and discharge, and shear stress, and looking at differences in water quality using such parameters such as ammonia, pH, temperature, and nitrates, to name a few. And then in late June, we collected and individually pit tagged 200 common mussels of the same species known as spikes. All of these were then moved to their new sites, the 3 experimental sites and the 1 control at 50 individuals at each site. There is still some work yet to be done for this project. This includes the resurvey of the mussels at the end of September. At the time of this recording, this work has not been done yet. During this resurvey, we will collect all mussels found and measure for changes in growth, mass, and look to see if they have survived the process or not. All living mussels will then be replaced into their sites and then once again resurveyed at the early June of 2021, where they will again be checked for changes in growth and mass and once again mortality. But why does this matter? As previously stated, freshwater mussels are very imperiled and provide very important ecosystem services such as habitat, stream stabilization, and food for many species. Translocation, as previously stated, is a pretty common practice used throughout the country by various agencies to help to conserve the remaining populations of mussels. The goal of this project is to help improve the translocation process here in Michigan to allow more mussels to survive the process and hopefully help to improve the translocation process in other states as well. I'd like to take a second to thank everyone who has helped me with my project so far, especially my advisor, Dr. Eric Snyder. In addition, I would like to thank Michigan Space Grant Consortium, Pierce Cedar Creek Institute, and the Grand Valley State University Graduate School for providing funding for this project, and I would like to take a second to thank you all for attending today's event.